in the department. So welcome to our Spring 2020 Symposium. We have Cherryle and Marisol, two soon-to-be GS graduates, who will be presenting their research findings to us today. For everyone in the audience, I'd like to give you just a little background on how the students got to this point. Our Department of Oceanography launched the GS program in 1998 with the generous support of the Poly Family Foundation. GS program is a vision and brainchild of Emeritus Professor Dr. Fred McKenzie, and it's structured around three core principles: the rigorous STEM curriculum, an integrated study of environment and sustainable related issues, and finally, and why we're here today, the faculty mentored research thesis requirement. So this research experience means that every GS student has to complete a faculty mentored research project, write a thesis, and present their results in a public forum. And so it's in recognition and celebration of this undergraduate research why we are here today. So before we get to the presenters, may I ask you as the audience several things. First off, please keep your microphones muted until the end of the presentation. Also, please do not comment in the chat window until the end of each of the presenters' presentation. After each presentation, if you would like, you can unmute your microphone for a few seconds to show your appreciation, and then please mute them again so that the presenters have an easier time fielding questions. And once we get to the question and answer portion of the presentation, if possible, if you can remember, maybe raise your hand or indicate in the chat window that you have a question, and I'll do my best to moderate so we have as orderly a process as possible. So the students have an easier time addressing your questions. Okay, so like I said, our presenters for today are Cherryle Heu and Marisol Plazas. And so I will now turn it over to Cherryle.
Okay, thank you, Dr. Goodry. Let me share my screen. I'm assuming that's okay. Okay. Can you see that?
Yes.
Okay, great. Hello, everyone. Again, my name is Cherryle Heu. Thank you so much for tuning in to my thesis presentation. My thesis is titled, Comparing Undergraduate Research Student versus Advisor Ratings of Student Performance Suggests Low Student Self-Efficacy, particularly among underrepresented students. So my thesis had two research questions. The first was, how do advisor assessments of undergraduate researchers compare with the student self-assessments? And the second was, are there any demographic differences? So the undergraduate researchers that was studied in this thesis was the SOAS Scholars Program. The SOAS Scholars Program had a total of 36 undergraduate researchers, and at the end of the program, an online survey was administered to the students as well as their advisors. Out of the 36 students, 30 students had responded to the survey as well as their advisors and gave us a response rate of 83%. So the students and the advisors, they were given a survey that was identical, and they were asked to rate the students' research skills and performance in these 10 areas listed here. And they were asked to rate on a 5-point scale from unsatisfactory to excellent for each item. So we had a total, we had 30 student advisor pairs and 10 survey items. So we had a total of 300 paired responses. So for data analysis, the survey responses were quantified to a numeric scale from 1 to 5. And then the mean difference was found by first finding the mean rating of the advisors and then subtracting the mean student ratings. Then to test for significance, a non-parametric permutation test was used. So a permutation test, this permutation test was conducted using a self-produced MATLAB code. What we chose this test based on the assumptions that we could make on our data. So again, the mean difference, D, was found using the mean advisor minus the mean student rating, and that was found to be 0.18. Since it's positive, it indicates that the advisor had rated on average higher than the student at a value of 0.18. So the permutation test was run by the code would take the 300 pairs and randomly select pairs to swap the student and advisor rating. So that means that when the scholar rated themselves a 4 and an advisor rated the student a 5, then the scholar value became a 5 and the advisor value became a 4. If the pair was not selected, then the values remained the same. So once the swapping was done, the mean difference was recalculated, and that was done for a total of 100,000 permutations. And the results of the 100,000 permutations are shown on this histogram here. And this red line indicates the 0.18 mean difference we found in our observed data. So using the observed data or observed point, the p-value was calculated empirically, and it was found to be 0.005. So this indicates that the SOAS scholars had significantly underrated their skills and performance when comparing to how the advisors rated them. The mean difference was again 0.18 and the p-value was 0.005. So these results can indicate or reflect low self-efficacy amongst the undergraduate students. So what is self-efficacy? Self-efficacy is a term that has been used in STEM a lot recently. It's defined as one's belief of their own abilities to perform a task successfully and overcome various challenges. If someone has low self-efficacy, they tend to underestimate their own skills and performance, and they're less likely to overcome challenges that they may face. And it can be used on a variety of scales as well. For example, it can be used on whether you'll do well on an exam or how well you'll succeed in your career in STEM. Studies have found that having a high level of self-efficacy can have a positive impact on your performance and persistence. And it's also found to be a better predictor of persistence in a STEM career than cognitive skills like research or your knowledge alone. So perhaps given the importance of self-efficacy, perhaps there needs to be, the SOAS Scholars Program needs to focus on building self-efficacy amongst or for their undergraduate students. So this leads to my second research question. Are there any demographic differences? So we looked at the students' gender and ethnicity. And so for the gender, a little over 50% of the students who had responded to the survey were female and the rest were male. And so the survey responses were separated based on the gender, and then the permutation test was run for both groups. We found that the mean difference for the female students was 0.24, which was much higher than the male students' mean difference, which was 0.09. The significance of the mean difference for the female students was also much higher than the male students. As for ethnicity, 50% of the students were Native Hawaiian and or Pacific Islander, and the rest were of these ethnicities here. What we did was we grouped the ethnicities based on whether they were indigenous or non-indigenous. And what we found was Native Hawaiian / Pacific Islander or NHPI students had a higher mean difference value of 0.24 compared to non-indigenous students who were 0.11. The p-value for NHPI students was also highly significant at 0.01. While non-indigenous students, the significance value was 0.18. It's important to note that for the four groups here, the difference or the mean difference was a positive value, indicating that the advisor had on average rated higher than the student for each group. However, those that are considered underrepresented in STEM, so the female and the NHPI students, had a higher mean difference and a higher significance in that. So that leads to my next concept called intersectionality, and that refers to when someone belongs in more than one underrepresented group. So for example, a student that is Native Hawaiian, Pacific Islander, and female, sorry. And so those students may go through what is called multiple discrimination, because those students may not only go through sexism, but also racism. So they tend to face more challenges than others. So as an intersectional analysis, I looked into 4 subgroups of the students based on whether the student was male or female and NHPI or non-NHPI. What was found was the mean difference for students that were female and NHPI had a value of 0.36, which was much higher than the other 3 subgroups. As you can see, the advisor mean rating was, the difference between the advisor mean rating and the student mean rating was very large compared to the rest of the subgroups. Also, the female NHPI students had a higher significance value of 0.02, while the rest of the subgroups did not have a significant value. So to summarize, the SOAS scholars overall had significantly underrated their skills and performance when compared to the advisors' ratings. The mean difference value was 0.18 and the p-value was 0.005. The female NHPI students, when doing the intersectionality analysis, we found that the female NHPI students had underrated themselves the most and the most significantly at a mean difference of 0.36 and a p-value of 0.02. So considering how the female NHPI students were the ones that had the highest mean difference and significance compared to the rest of these subgroups, it suggests that perhaps the female NHPI students were the ones that were driving the results of the first analysis done when we did the 300 pairs. So the underrating of the 300 pairs was mainly due to the female NHPI students underrating themselves. So again, this may reflect lower self-efficacy, especially for the female NHPI students. So going back to and considering how self-efficacy has been found to be a large factor in one's success in STEM, perhaps undergraduate research programs could benefit from building self-efficacy intentionally alongside cognitive skills. This could result in a higher retention rate in STEM majors, as well as if we focus more on underrepresented students, it could result in a more diverse workforce. So how can these programs build self-efficacy? Arthur Bandura is a psychologist that coined the term self-efficacy, and he highlighted 2 main factors that contributes to one's self-efficacy. The first being mastery experiences, which involves when someone faces a challenge and overcomes it kind of continuously in a cycle where, so for example, if an instructor were to provide a student the opportunity to troubleshoot in field activities continuously and let them overcome it one by one, they will gain from that and gain self-efficacy. So the next factor is vicarious experiences, and that involves when someone observes the success of someone else, that particularly when they can identify with that person. So for example, having a diverse faculty and peer mentor that students can work with, if they're able to look up to them and if they can see someone similar to themselves with the success that they have, they may be able to gain self-efficacy. And in a geoscience research program that was done by Quartz, she found that by incorporating these experiences into the research program, the students were more likely to persist in their STEM careers. So there are a few limitations to my study. The first is the quantification of the Likert scale. Quantifying the unsatisfactory to excellent scale to a numerical scale from 1 to 5 may not necessarily reflect the response of the students and the advisors. Second, the survey was not externally validated, meaning this survey, though we interpreted it to mean low self-efficacy, it could potentially have other implications as well. So to address those issues, what could be done is what's called a mixed methods analysis. So instead of focusing on only quantitative analyses, incorporating qualitative factors by, for example, interviewing students can perhaps, perhaps that would let us look into the level of self-efficacy of the students even further. Also, having a larger sample size, for example, surveying the undergraduate programs or more of them could influence the results and also further the demographic analyses by, for example, looking into the underrating of each individual ethnicity instead of lumping them all together. Thank you so much, and I will welcome any questions you have.
Okay. Great job, Cherryle. And she will now take questions. Let's see, so are you able to see Rosie's question?
Yes.
Okay. Right.
Okay, sure. So specific strategies so the first question was, what specific strategies do you suggest SOAS employ to help improve the self-efficacy of intersectional students? So again, looking back here, so I highly suggest, especially for professors, to look into the Bandura and Quartz papers. It's especially important to incorporate self-efficacy into research programs like REUs, where you don't only develop the cognitive skills in the research program, but also develop the self-efficacy. I'd say, again, troubleshooting for field activities, or just being aware of this, and not only focusing on the environmental science aspect, but being more conscious about incorporating self-efficacy building will make a huge change, or will make a big impact on the students. The second question was, have you looked at any other interactional parameters? Unfortunately not. We did not have a large enough sample size to do this, so definitely that's something that can be done in the future works. Let's see, and the third was, have any other experiences other than mastery or vicarious be assessed, such as cultural-based methodologies? So this slide, or my research for how to build self-efficacy was heavily done through Bandura's papers, because he's the one that kind of dedicated his career into self-efficacy. I'm not sure if there are any cultural-based methodologies. I feel like that's extremely helpful, especially for students in Hawaii, but I will have to look into that. Hope that answered your questions.
Great.
Thank you.
Great. I know Jim Patimra raised his hand. Jim?
Yeah, hi, thanks. Hey, Cherryle. So I was wondering, you did the study based on from the student side, but did you also look at the makeup of on the advisor side? I mean, are the advisors presumably more homogeneous in terms of where they come from and what their ethnic background is, and maybe that with your results, are you able to say if that matters?
Okay, that's a good question. So we only looked at the ethnicities of the students. One important thing to note is that the surveys were done anonymously. So we had the code, and we're able to ask for the ethnicities and the gender for the students, but we did not specify for the advisors. I think one thing that could be looked into if we had a larger sample size was perhaps if, for example, a student and mentor had similar background or similar ethnicity and whatnot, looking into how those students underrate themselves compared to other peers may be something really interesting to look at.
Great, thank you.
But not yet.
I have a question. Hear me?
Sure. Yeah.
Yeah. Okay. You can hear me, right, Cherryle?
Yes, I just wasn't sure who it was.
Yeah. Sorry. I think fantastic study. Very interesting results, and it kind of begs the question: being local, right? Being a SOAS GS student, having gone through our curriculum, academics, and then the co-curricular research experience, and now done this study, do you have some concrete ideas or suggestions on what we could do with our, not just GS, but with SOAS programs overall?
There's a lot that can be done, I'd say. Although I focused on the research programs, I believe that departments and classes can also somehow incorporate this, I can get back to you on that. I do have the resources, but I have not compiled them for this presentation. I only kind of focus on the research programs, not necessarily the departments.
Thank you. Do we have any other questions? Great job, Cherryle. Well done.
Thank you so much.
Okay.
Okay.
So that was the conclusion of our first speaker. Now we will transition to Marisol Plazas. So Marisol, if you can share your screen.
Sure thing. Good. Sorry, it minimized and I just lost it. There we go. Okay. Hi, everybody. Thanks for coming out, supporting your gal. So this is the exciting world of deep sea mineral composition found in blue mud samples taken from the Mariana Forearc aboard IODP Expedition 366, presented by yours truly and advised by Patty Fryer. So I've quickly outlined our key points today for you guys. First, we're gonna go through a bit of geological background, catch you guys up to speed. Gonna dive into the methods because that is definitely how we did things. Some analyses, and finally the best part, the conclusion, not only because we're gonna be done with this, but because it's the most interesting part. So within this study, there were two goals. The IODP expedition goal, which was to recover drill core samples from 3 active serpentine mud volcanoes on the Mariana Forearc. Those 3 were the Asut Tesoro, the Fantagisnia, and the Inizao Seamounts. So once those samples arrived here at UH, my goal was to then determine and compare those minerals from 2 of the sampled serpentinite mud volcanoes. Those 2 were the Fantagisnia and the Inizao Seamounts. As you can see here, in purple. So my whole thing behind this was to identify any differences between mineral composition of serpentinite mud volcanoes that erupted long ago versus what erupted more recently. This could indicate a change in processes within time. And so just to be clear, the Fantagisnia Seamount is where majority of the old mud flows were found, and the Inizao Seamount is where the more recent mud flows were found. So we did this to compare and see a change in processes over time. Minerals form under specific conditions to estimate indirectly the environmental conditions, such as temperature and pressure, that are within and surround these mud volcanoes, which may shed light on microbiota origins. So just to give you guys a better look at it, these are the 10 cores we gathered our 11 samples from. The yellow boxes are to indicate where on each core we gathered our samples from. Unfortunately, they weren't present for the photos, but it does give you a great idea of what the samples look like in colorization. So we have Group A, which represents the more recently erupted summit samples. Group B indicates deep edge samples, and Group C indicates the slightly shallow deep edge samples. Now, if you're kind of curious and happen to notice why this sample here has a more muddy, earthy look to it, we'll get to that. So hold on to your horses, you guys. And to give you guys a little bit more geological background, this figure shows a cross-section of the Mariana Forearc. As you can see, that the plate, the Pacific plate, is overriding the, you can see that the Mariana Forearc is overlying the Pacific plate. As the Pacific plate subducts, it releases water and other fluids from the lithosphere. The Mariana Forearc begins to stretch and create deep faults. As the fluids rise up into the fractures, through zones here. It reacts with the mantle and hydrates ground up mantle rocks to form serpentine. Depending on the temperature range, a specific form of serpentine can form: lizardite, integrite, or chrysotile. The newly formed mud flows are then erupted onto the seafloor, and they create the largest mud volcanoes on Earth, which I had the pleasure of studying. So just again to emphasize the serpentinite phases, that chrysotile and integrite up close. Thankfully, that's something we had in our bag of tricks to use, but we kind of saw more or less lizardite throughout majority of the samples. This is more or less due to the temperature ranges that they form in. I can't give you specific numbers because nature, it always changes, but it is the most common. Chrysotile, we saw a little bit more of, which is another second to most common. And integrite, unfortunately, we did not see in our samples, but maybe in future samples that others drill up later on. So this one looks a little scary, but we're gonna walk through it together. It's really easy when we break it down. So the serpentine formation process is important to understand to know the likelihood of microorganism conditions needed to thrive in and around the serpentine mud volcano. So pure olivine, magnesium olivine, is a major mantle mineral, which reacts with water to form serpentine and brucite. So as you can see here, we have our olivine, our water, our serpentine, and our brucite. This is typical, but for our samples, olivine in the Mariana Forearc mantle contains about 10% moles of iron. So when this iron olivine reacts with water, it oxidizes, forms magnetite, and liberates hydrogen. The hydrogen is extremely important for microbial metabolism. This is why serpentinization environments are thought to be likely for the origin of life. So, with all this new information I just presented to you guys and hopefully understood so far, we continue to question what are the mineral compositions of the mud samples. So, to do this, we used an X-ray diffraction, also known as an XRD. It gives information about crystal structures to identify minerals. So I want you guys to try to remember this image here. So as you can see, there's one arm here and another arm here, and a circle tray down here. So again, we have our arm here, our other arm here, and our circular tray here. So what we did was we grinded the sample to 63 microns. So for those of you who don't really know that size, it is like very small grains that look like dust almost, extremely small. Packed it into a clear-like puck and placed it into this disc here. And so after we did that process, we fired up the XRD, and the way it functions is pretty simple. There's an X-ray source that is emitted from this arm that hits the sample, which is sitting in this puck. And then the X-ray source bounces off the crystal structures found in the sample into the detector, which typically remains at a 2 theta angle. The detector then reads the intensity of the sample, or it also gives the count number of the sample. And so after this process is done, we get scans that look like this. So another scary looking one, but we're gonna walk through it. And we're gonna recap. So in the beginning, we had 10 cores, one of which we sampled twice. So we ended up with 11 samples and 11 ARC-SRD scans, which you see here fell within 4 groups. These scans were created by an analytical software called DFRACT-EVA. It produces really small labels, but all you really need to know is that they show angles 2 theta increasing to the right, and the counts keep increasing upward on the y-axis for each group. You can see here the reason why the scans were split up into their respective groups is due to their similarities. We use group A as our baseline since it had the least amount of peaks throughout their scans. But the thing to note within this group is that even though there are 4 of them, they came from 2 different seamounts, the Fantagisnia and the Unizao. So this is kind of significant. But if you look at group B, you can see that the patterns are very identical. But there's a small peak that forms off the shoulder off of each one of the 3. Another thing to note, though, is for group B, they're also from 2 different seamounts, the Unizao and the Fantagisnia. Again, the similarities are kind of popping out at us. Group C, though, are not from 2 different seamounts. They came from the same one. But you can obviously see the similarities between group A, B, and C, despite that group C had a few more peaks coming out that weren't present in group B, but were present in group A. Well, not present in group A, but continuing on. So lastly, if you already took a great look at it, you probably already noticed that group D comprises of 1 sample, and that it's the most unsimilar sample to the rest of them. So this is because it comes from beneath the serpentinite mudflows of the Fantagisnia seamount. So again, this is where it came from. This is our red Earth-like sample, and it came from underneath the seamount. So the pieces are starting to fit together at this point. But yet another scary table. We're gonna get through this one, you guys. So this table shows the dominant, present, and possibly present minerals within the 4 groups sampled from the Mariana Fork. Across all 11 samples, we found mantle minerals, secondary alterations of mantle minerals, volcanic ash, and because we cored these from underneath the ocean, seawater. So from all of this so far, we discovered that the most common serpentine mineral was lizardite in 10 of the 11 samples. Olivine, halite, and calcite were prominent minerals in groups A, B, and C. Group D's diversity was different because it clearly represents sediments underlying the flank of the Fantagisnia seamount. Some of the seamount sediment was introduced as a result of contribution of ash from explosive volcanic events from the nearby Mariana Island arc volcanoes. Because feldspar and pyroxene were dominant in the sample, these minerals show that Group D consists of different minerals from groups A, B, and C, and are consistent with pelagic sediments found on the Mariana Fork, which likely represent volcanic ashfall, submarine landslides, possibly dust from Asia, and microorganism remains. But as for sample Group D, there's much more work to be done in identifying it thoroughly for future samples. So the great Kaplui. From all our newfound knowledge, we can conclude that the mineralogy hasn't changed very much except where Group D was found, and that the mineralogy has not changed greatly from early to late eruptions due to the samples being similar at both seamounts. The mud formation processes have not changed greatly between the 2 sites, and such deposits occur worldwide throughout Earth's history, and could likely be an environment for the origin of life. Lastly, there was a lot of talk about Group D, but as this is the only sample of its kind, we can only speculate the meaning behind it. If the origin of life is related to serpentinization, then we can expect that normal volcanic ash deposits on the surface of the Earth might be a good environment for the origin of life. And if eventually future expeditions are conducted, we can find evidence for a difference in microbial communities within the underlying ash. And with that, I would like to thank my friends, family, and honestly, everyone just involved in my life. So thank you for that. I should.
Great job, Marisol.
Thank you.
Okay.
Bring that back up.
Yeah, now we go to questions. And so, yeah, if you want to share, there you go. Yeah, so great talk. So let's see if we have some questions from the audience. Let's see, Jim Patimra.
Yeah, hey Marisol, nice job. I'm curious, maybe you said at the beginning, but can you give an idea about the age of these minerals that you're looking at? And I know probably everybody's focusing on D because it was so extraordinary, but you mentioned that it possibly could be Asian dust, and I'm wondering if you have an idea, if it was Asian dust, how old would that Asian dust be?
Okay, so for as the aging of these minerals, or likely these cores, there isn't much to be told about the ages. It's slipping my mind at this very second as to why. But if I had to take a gander at it, and obviously this is not a proper time frame of it, it would likely be before humans, maybe. I'm honestly not aware of the time frame.
I mean, if you really are measuring Asian dust and it's very old, then you might be able to, you know, make some sort of statement about paleoclimate and how the Asian dust actually got there X thousands of years ago, whatever that might have been.
Yeah,
Okay,
yeah.
thanks.
Yeah, no worries.
Okay, so Marisol, can you see the question from Malie in the chat window?
Oh, yeah. Hey, can you use your serpentinite muds to look at the composition of fluids derived from the Pacific plate? Well, Emily, likely, yes. Yes, you can do that. Because that's kind of what we did. Basically.
Okay. I have a question, Marisol.
Yeah.
Okay, so if so based on all the work that you did, what would be your suggestion if someone were to take up where you are left off? What could be some of their next steps?
Firstly, have at least a minor in geology. But next steps in all seriousness would be A, yes, know your stuff as best as possible. B, try to see if you can get similar samples for group D, and maybe it'd be pretty cool to see some dating, honestly, if that's at all likely. But just kind of know your stuff is the biggest one.
Do you know if based off of this unique group D sample, if that is, do you or your mentor, you feel like that's sort of strong enough to go out and try to get an additional cruise to get more of those type of samples? Is that in the works, or?
I mean, it's not in the works as far as I'm aware, but it definitely should be something that should continue to be researched. Just for example, we all know how little the oceans have been actually researched, and I know there's a ton of things being done to, you know, get the layout of the underwater world, but unfortunately, because it is so deep, it is difficult to go down and recover cores like these. However, I think within time, hopefully not in 5 decades, but I think we're coming closer and closer to getting the technology needed to recover more samples like this. That's the biggest issue is the fact that it's so deep into the trench, or well, not into it, but like off the fork. But that it's still a pretty long ways down pressure wise for a lot of machinery that we currently have in today's world to recover samples like this. But hopefully in the future, there'll be more that can be done to do that.
Hey, Patty, you had a comment to make, maybe?
Yeah, that was great, Marisol. You did mention that future work was needed on those sediments from underneath the mud volcano. And people who are out on the cruise are doing research on the microorganisms that were collected in that material. So haven't heard anybody come back with definitive dates or even mineralogical analyses that might indicate that there were materials, dust from China, for instance, but this was something that people were funded to do, and they have a few more months to complete their post-cruise analysis. So yeah, it very may well be that we'll be able to get some ages on that material and some determinations of the origin of them. Whoops, that's my dog, sorry. I'll mute myself again.
Thank you, Patty. Okay, are there any other questions for Marisol? Okay, if everyone could unmute their mics and give me a hand for both presenters today.
Great job.
Oh, Marisol. Good for you. Marisol.
Good, Marisol. Congratulations.
Well done.
Good work. And
Good work, both.
I'm very proud of you, muñequita.
Thanks, Mom.
Buen trabajo. Well done.
Good job, Cherryle.
Good job to you too. Thank you, everyone.
Thank you for everyone for attending, and have a great weekend. Aloha.
Thank you.
Aloha.
Aloha.
Hi, thank you.
Bye.